This is a video on B2 Edexcel GCSE Biology or Additional Science, Topic 2, Organisms and Energy. So it starts to do with respiration, moves quickly onto photosynthesis, and it's the middle topic of the three topics in B2 Biology. So let's get started. So first idea that comes up is respiration. Now respiration is a key idea, I would say, in the area of biology. So respiration is the process by which all living organisms release energy from organic molecules like glucose, so sugars essentially. This energy is used to make larger molecules, so proteins, also can be used to contract muscles and also maintain a steady body temperature, which is part of a homeostasis, thermoregulation, which should know for B1. There's two types of respiration. The main one of which is aerobic. This is what we do pretty much all the time. It's where you take glucose and oxygen, and from that creates carbon dioxide and water. Obviously, and the aforementioned energy is released as well. So the body gets glucose from food, oxygen comes from the air. Glucose and oxygen are transported around the bloodstream, they diffuse into cells, and diffusion does come up. Carbon dioxide is produced as well via respiration, and that diffuses out of the cells. So oxygen and glucose diffuse into the cells, carbon dioxide diffuses out. Water is also produced, and that generally stays within cells. If you're exercising, you will respire more because you need more energy, mainly from muscular contraction. So that means the oxygen needs to get to your cells. So that increases your breathing rate and increases your heart rate, so that your heart beats faster, pumps more oxygenated blood around your body, therefore it's going to get to your cells quicker. This means the rate of diffusion at the cellular level must increase as you faster diffusion, and therefore more carbon dioxide can leave if more oxygen glucose can get in. So you can actually calculate your cardiac output, so that's the volume of blood your heart pumps out per minute, by doing the heart rate times by the stroke volume. Vigorous exercise can lead to your body not being able to have enough oxygen. So this leads to anaerobic respiration. So anaerobic respiration when you don't have enough oxygen. So what happens here is the glucose goes straight into lactic acid. Now lactic acid is what causes cramps essentially. Less energy is released than before. The glucose is not fully oxidized, therefore less energy is released. Oops. Right, so a lactic acid buildup can lead to cramp essentially. So when you stop exercising after anaerobic respiration, you're in oxygen debt. Your body needs oxygen, and it uses that oxygen to break down the lactic acid. That's why you pant after exercising heavily. So the amount of oxygen required is called the excess post-exercise oxygen consumption, which is the EPOC. This means that you still breathe quickly after exercise, and your heart rate will stay high for a while until all that toxic lactic acid is broken down into carbon dioxide and water. So you can investigate the effect of exercise on your breathing and heart rate, performing different types of exercise and measuring your pulse rate per minute. Right, diffusion. So there's a very clear definition for diffusion here, which has been underlined. It's the process by which particles move from an area of high concentration to an area of lower concentration. So it's down the concentration gradient, from high to low, down the concentration gradient. While respiration is taking place in cells, oxygen and glucose are being used up. This results in less oxygen and glucose being in the cells, and there's a higher concentration in the blood. Therefore, it goes down the concentration gradient from the blood into the cells. So that's how it moves from the capillaries into the cells. The capillaries take carbon dioxide from the cells because the cells have a higher carbon dioxide concentration than the capillaries. The greater the difference in concentration, the faster the rate of diffusion. So if there's a big difference in concentration, the diffusion happens very, very quickly. This takes us on to photosynthesis. If you look carefully at the equation of photosynthesis, it's the equation for respiration backwards, but with sunlight and chlorophyll added. So it's carbon dioxide plus water, and sunlight and chlorophyll are above and below the arrow, and that produces glucose and oxygen. You need to know this equation. There are four things that are required for photosynthesis: carbon dioxide, water, light and chlorophyll. Carbon dioxide, that enters via the stomata, water taken in via the roots, light is usually from the sun but it can be artificial as well, and chlorophyll, that absorbs that sunlight energy and that is used to convert water and carbon dioxide into glucose. So that energy is used to do that. So there are three things that affect the rate at which photosynthesis occurs. Okay, so there are three graphs that show you these, and they're shown below. The diagram over here is an experiment which can be used to sort of investigate the rate of photosynthesis. As photosynthesis increases, more bubbles are released. Therefore, the level of gas in the inverted measuring cylinder will increase. So this level of gas will go down, down, down as more oxygen is produced. So let's have a look at these graphs. Right now they're missing off there, but I'll draw them for you somewhere. Apologies for the missing. I think if I says it, I move this down, they might magically come back. I think it's because they're hiding. Yeah, there they are. Let me see if I can move it down here. Sorry, give me 2 seconds. There we go. Okay, right. So they're the graphs. Apologies for your workbooks not having those graphs, but if you're watching this video, you'll figure it out. So here's figure 1. This shows the rate of photosynthesis changing with light intensity. So you can see that as light intensity increases, the rate of photosynthesis increases. It increases to a point because remember, light is a limiting factor. Over here, it's taking as much light as it can. It can't take any more, so the rate of photosynthesis levels off. It can't take any more light to make the photosynthesis happen faster. You need to change one of the other limiting factors now. Carbon dioxide concentration has the same graph. Again, at this point it's saturated. It cannot take any more carbon dioxide. You need to change another limiting factor to change the shape of the graph to make it happen faster. Temperature is a limiting factor, so it's different to the other two. It increases to a point and it decreases, generally about 45 degrees, that point is. Now remember, above this would be enzymes denaturing. That's the reason why the rate of photosynthesis is decreasing. Yet again, enzymes rule this reaction. Right. The amount of light a leaf receives can be controlled by using a lamp. So that lamp can move closer or further away. The amount of CO2 the plant receives can be controlled by adding sodium hydrocarbonate to water. And the best way to control temperature is by placing a leaf in a water bath. So you can investigate each of those three limiting factors. So these leaves, they're actually adapted to maximise photosynthesis. So they've got a large surface area to take in a lot of light. They contain chlorophyll and chloroplasts to absorb light energy, and they also have many stomata at the bottom of a gas exchange. That's carbon dioxide and oxygen, and water vapour as well. The stomata, or stoma for singular, are small holes on the underside of the leaves. They open and close to get gases in and out. So we've talked about gases moving. What about water? Well, water moving has its own special name. That's called osmosis. You might have come across the idea of osmosis before. Osmosis is a diffusion of water. It's the movement of water from an area, a high concentration of water, to a low concentration of water. It occurs through something called a semi-permeable membrane, which allows certain size particles through, but doesn't allow others. It allows the water particles through, but larger particles don't go through. It's a bit like a colander or a sieve. So that's how water gets in and out of cells. Osmosis occurs as there is a difference in concentration inside and outside the cell. The more water there is, the substance, the more dilute it is. More dilute means less concentrated. So osmosis is the process by which root hairs take in water. So root hairs cells have a large surface area, which means a lot of osmosis can take place. So you can do a really simple experiment with this to do with potato chips. So you take potato chips and place them in water or a sugar solution. The mass of the chip is measured before and afterwards. And if the mass is increased, then water is moved into the cell. There's another process by which minerals move around, and that's called active transport. Active transport is how minerals move within the plant. So root hair cells absorb minerals from the soil, and they need to go against the concentration gradient. They need to go from areas of low concentration of this mineral to higher concentrations. So this is called active transport. It requires the energy from respiration to do this, to help pull the minerals against the concentration gradient. You need this process for growth, because it can't get minerals to where they need to be. Transpiration is caused by the evaporation of and diffusion of water from inside the leaves. This process leads to a slight shortage of water inside the plant, which leads to the plant draw more water in from the xylem. That's essentially the part of the plant that transports water and minerals around the plant. So that leads to more water being drawn in from the roots. So without transpiration, without that evaporation and diffusion of water taking place, more water will not be taken in from the roots. This ensures that there's a constant supply of water for photosynthesis. Phloem moves sugars, while the xylem moves water and minerals. Right, so the last little bit that we need to look at, I'm just going to confirm this is the last bit, yes it is sampling and how we actually sample the distribution of organisms. So there are many ways to do that. The first one is pootas. This is a poota here. Insects are put into a container through the metal tube. Okay, you inhale through the flexible tube, and the fineness prevents you actually inhaling any of the organisms in here. So you can actually suck organisms into this poota, into this container, by sucking them on this tube over here. There's a mesh over here which stops the insects actually getting through. This is what you use to collect ground insects. You can suck the shorter tube, or the flexible tube, sorry, and place the longer end tube, this one over here, where technically this one's longer and that one's shorter. And you compare how many insects you find in a different area of the land or in the same amount of time. You also can use pond nets or sweep nets. You've seen these before, they look like this. They're used to collect animals in long grass. Sweep net is lined with a strong cloth. You can use to collect insects and spiders. It's easy to stand in the sample, sweep the net once from left to right across the grass, then quickly sweep the net up and turn the insects out into a container and count them. Obviously you can repeat this to see what you get in the pond, choose that and do it in a similar way. Pitfall traps. It's like essentially a little trap with a trap, and inside the trap you've got these deep-sided containers that insects can fall into and can't get out of. So you can see what sort of organisms would be in an area. So it's partially open at the top. You leave the trap overnight, insects fall in, can't get out, so you can compare the different insects you get in there. The final method that we're going to discuss is a quadratic. So it's a square frame enclosing a known area. This is used to compare how common an organism is in two different areas. So you take this and just throw it out into the grass or whichever area you're examining, and then you count how many you find in certain areas. So the quadratic is placed on the ground at a random point within the first area. Count all the organisms that you're interested in within the quadratic. Repeat these steps and work out a mean. You can repeat for different sample areas, or you can compare your means. So you could count how many and one and then times that by whatever the size is to figure out what it would be of the area, or you could take it from 3 and then calculate a mean of the 3, whatever you wish. And we also need to find out the effect of different environmental factors. So temperature can be measured using a thermometer, or light can be measured using light intensity meters, and you could and indicate liquid can be used to test pH. So you could sort of test this in different pHs and different lights, intensities and different temperatures to see what's going on there. And that is the end of B2 topic 2 for Edexcel GCSE chemistry.